All right, hello everyone. My name is Anne Bertolotti. I'm a program leader at the MRC Laboratory of Molecular Biology in Cambridge in the UK. And today I'm here to try and hope to perk your interest on protein phosphatases. These are a class of enzymes that control pretty much every aspect of life, and yet they've been very much neglected in the past. They were thought to be unselective and undruggable, but I will tell you in a set of 3 talks today that this has changed recently. But before I get there, I'd like to give you a short biography as a means of an introduction. And the point I would like to make with this is that not only science is great fun, but the careers that come with it are also very exciting too. I trained in Strasbourg in the eastern part of France as a biochemist, and I then moved to New York. This is where, as a postdoc, I began my work on the cellular defense against misfolded proteins. I then set up my lab for a few years, initially in Paris, until I moved to Cambridge, where I have been since 2006. So most of the work I'm going to discuss today has been carried out in Cambridge, although some parts started in Paris. So, as I said, I will give 3 talks today. The first will be this talk, where I'll give you my own perspective on the historical discoveries of protein phosphatases. In the second talk, I'll share with you some of the work that was carried on in my own lab, where we discovered how we can selectively inhibit a phosphatase. And in the third talk, I will outline for you a set of principles that can help everyone, I hope, studying phosphatases and identify selective phosphatase inhibitors. And in the background of all this, I'll illustrate for you the power and the benefit of selective phosphatase inhibition in the context of neurodegenerative diseases. But first, I want to give you a general introduction to make sure we all start on the same page. So all the information that is required to sustain life is encoded in our DNA, and then this information needs to be transcribed into messenger RNAs that then will be translated into proteins. And proteins are will execute the vast majority of cellular function. So a protein is a sequence of up to 20 different amino acids, and what determines a protein function is not only the sequence of amino acids, but also the way a protein folds. And protein folding is very important, and in addition, failure to fold proteins leads to their accumulation, and accumulation of misfolded proteins is a huge problem for cells and organisms, as I will touch on in my second and third talk, and I'll tell you that accumulation of misfolded proteins causes neurodegenerative diseases. But in today's talk, I will focus on another element which is very important in controlling the function of proteins, and this occurs after a protein has been made, and therefore this is called post-translational modifications. And why is this important? Well, life depends on our ability to adapt quickly to the many changes in our external or internal environment that occurs all the time. And the way we adapt to changes is by controlling the presence or the absence of proteins, which carry out, as I said, the function that are important for our cells and our organisms. So to control the levels of proteins, we have controls at every step of the flow of information in the cell. Transcription can be controlled, translation can be controlled, but we can also control protein abundance by controlling its degradation. So all these steps are very important, but it all takes a lot of time. So post-translational modification are really a quick way to add a little modification to a protein, and in this way turn on or off its function. So there are about 200 different post-translational modifications that have been identified so far. But today in this talk, I will focus arguably the most prevalent one, which affects a very large number of proteins in our cells, and in this way controls pretty much all aspects of life. And this modification is the phosphorylation of proteins. So I'm going to start with giving you an overview of the historical discoveries in phosphatase biology, and I hope you'll find that fascinating. I certainly do. So I'm going to start in 1943 and take you in the lab of Carl and Gertie Corries, who were then studying enzymes important for glycogen metabolism, and particularly they were interested in an enzyme called phosphorylase, which is an enzyme that catalyzes the breakdown of glycogen, so a very important enzyme. And they noted that this enzyme can exist in two forms, one called phosphorylase B, which is actually inactive, which differ from the active form, phosphorylase A, in that it has a little addition of something that they realize is not a protein, but they called it a prosthetic group. It's a non-protein group. And they realized rightly that this group is actually tightly bound to the protein. It's actually covalently bound. And to be removed, it needed an enzyme that is present in a muscle extract. And this enzyme, they call it PR for prosthetic group removing enzyme. And you'll see later that this turned out to be, in fact, a phosphatase. So this was an important discovery. They realized in this way that an enzyme, the activity of an enzyme can be controlled, and for this discovery, they won a Nobel Prize. But the nature of this small modification took actually many more years until it was discovered. And I'm going to tell you about this in the next slide. So the discovery of protein kinase took actually, as often in science, a very tortuous route. And this began with Ed Krebs, who was a postdoc with the Corries. And when he moved away and started his own lab, he actually failed repeatedly to produce the active phosphorylase A that he used to produce in the Corries lab. And instead of giving up in the face of failure, he actually teamed up with Ed Fisher, and together they converted this problem in, as you will see, a prize-winning discovery. So what they did, they actually scrutinized the different steps of the purification as they were doing it in their own lab, and compared that with rigor and diligence to the purification procedures that the Corries had established. And they realized that there was a key step required to purify active phosphorylase A, and this key step involved filtration of the tissue homogenate through paper filters. And they went as far as burning the paper filters and added that and realized that this was sufficient to recapitulate the effect of the filtration, leading them to realize that the active component was actually an inorganic component. And this was, in fact, a metal ion. And in this way, they realized that they can convert their failure, namely the inactive phosphorylase, phosphorylase B, into an active enzyme by incubating it with metal ion, ATP, and a cell lysate. And this was the first discovery of protein phosphorylation. And this is a very important discovery. This is a discovery of a kinase, in fact, and it's a very important discovery because not only protein phosphorylation controls in this way the activity of phosphorylase A, but as I will show you later, it also controls the activity of many enzymes in the cell. So this is a discovery of very broad significance, and we are very lucky that Ed Fisher himself gave an iBiology talk, and I would like to encourage you to watch it. It's really a fantastic way to learn more about the early days about protein phosphorylation. So what is protein phosphorylation? It's a remarkably simple modification of proteins. It consists of the addition of a phosphate group on residues predominantly serine residues, but also threonine, also tyrosine and some other residues. And this reaction is catalyzed by kinases, which add a phosphate group to proteins, and the removal of the phosphate is catalyzed by phosphatase. And in this way, the activity of a protein can be switched on and off. So how does it works? Well, first let me tell you that this modification has a broad range of effects in the cells because it affects a very, very large number of proteins. And I've taken these numbers out of recent reviews from Matthias Mann, showing that if you can identify 14,000 proteins in a cell lysate, you may find as many as 1 million phosphorylated peptides. So there's really, tell you that protein phosphorylation is widespread and modifies the activity of many, many proteins in the cell, and in this way controls virtually all aspects of life. So what is the consequence of protein phosphorylation? So this is very interesting. It's a small modification, and as you will see, it's very versatile. Why is this? Well, if you add a phosphate group to a protein, this it's a small modification, but yet in the context of the size of a protein and amino acid, it's a bulky modification which will perturb the way a protein fold, and it will also add a charge because the phosphate group is highly charged, and in this way this also impacts on the way a protein folds. And because of that, this modification is highly versatile. It impacts on the function of protein in virtually every possible way one can think of. Protein phosphorylation has been reported to alter protein stability. It can target protein to degradation. It's also a way to control protein localization, and obviously it also impacts on protein-protein interaction. So if you think about a small protein-protein interaction interface, if you add a phosphate group, obviously this is going to block the way two proteins can interact, and conversely, if you add a phosphate group at the surface of a protein, this may create a new interaction site for new interactions. So a highly versatile modification that controls protein in virtually every possible way. And so the way protein phosphorylation was initially described by Fisher and Krebs is a switch that can control the activity of a protein on and off, and this can be either way. But actually, if you think about the fact that there are many molecules of a given protein in the cell, protein phosphorylation can vary all the way from 0% to 100%. And so in this way, I think that protein phosphorylation is perhaps more like acting like a rheostat that can finely tune the activity of proteins to precisely match the demand. So a fine way of controlling the activity of a protein to control, as I said, all aspects of life. So with that, I'll now take you through what I think are key and very influential discoveries that have shaped the way people think about phosphatases. So here I'm going to take you in 1973 with another serendipitous finding, which came with a group trying to purify the phosphatase that the Corey had described with their established protocol. But here came a mistake, and through the mistake came something interesting that had quite some influence on the phosphatase field. So in this paper, the author reports that when they purify the phosphorylase phosphatase, here they added ammonium sulfate and ethanol as a precipitation step. And in this way, they precipitated the phosphatase, but it turns out that by this way, they recovered an enzyme which was no less than 43 times more active than the protein that had been purified by the Cories. And this came about with the conversion of the enzyme that was normally present in multiple molecular weight species, but through the precipitation procedure, this converted the phosphorylase phosphatase into one single molecular weight species of 30 kilodalton. And I want now to summarize this historical paper for you in a cartoon format, because this is a very important finding that had a big influence on the phosphatase research subsequently, as you will hear during this talk. So in cells, phosphatase is present in multiple molecular weight species, and this is because it is bound to one amongst many different other cellular protein. And through the harsh purification procedure consisting of mixing ammonium sulfate and ethanol, the phosphatase precipitates, it forms a sort of gloops in the tube, falls out of solution, and out of that one can recover an active phosphorylase phosphatase. It's super active, but it's no longer bound to its cellular cofactor. So one needs to keep this in mind. This is a purified protein, which is very different from the cellular protein. So following this, the following year, in 1995, came the discovery of an inhibitor, inhibitor 1, and this inhibitor was instrumental in enabling the characterization of other phosphatases that have been discovered following the discovery of the PR enzyme that then was called by the group of Philip Cohen, who classified and discovered some of these other phosphatases, protein phosphatase 1. So more phosphatases were discovered, but only protein phosphatase 1 was inhibitable by inhibitor 1. And today, turns out that the phosphatase family has grown. PP1 is the founding member of a large family of phosphatases that is called the PPP family. Now, these enzymes arose from a common ancestor, and because of this, they share a common mechanism, and which is, has been elucidated here in 1995 by two groups. So in all PPP, there's in the active site 6 conserved amino acids, and they're very important in binding 2 metal ions, which are shown here in blue on this slide. And these metal ions are important because they will activate a water molecule which will then be able to engage in a nucleophilic attack and attack the phosphate group. And this is how dephosphorylation is carried out by this class of enzymes. Following this, following the discovery of inhibitor 1 and more phosphatases, people were looking at phosphatase specificity and how many substrates the different phosphatases were able to dephosphorylate. And initially, the lab of Philip Cohen up in Dundee described the fact that protein phosphatase 1 can actually dephosphorylate many enzymes of glycogen, involved in glycogen metabolism. And this was a first step leading to the idea that phosphatases are not selective. They can dephosphorylate many proteins if they come nearby, and that's how they work. And so the idea that phosphatases were not selective gained gravitas with the discovery that of many genes encoding serine threonine kinases. 400 genes encoding serine threonine kinases were discovered, whilst only about 40 genes encoding serine threonine phosphatases were known. So these numbers have changed a little bit recently. I'm taking here the numbers from a review from Manning, 500 or so kinases for 100 phosphatases. So the problem remained, right? We have 5 times more kinases than phosphatases. And that, together with the fact that phosphatases, the purified enzyme, remember, not the cellular enzyme, the purified enzyme, in the test tube can dephosphorylate many substrates, this idea, together with the fact that we have many more kinases than phosphatases, led to the notion that phosphatases are not selective. And this was a problem for phosphatase biology because who wants to study enzymes that are not selective? Who cares? The catalytic mechanism had been elucidated, I told you, in 1995, and if there's no level of regulation there, not so many groups were interested in studying this. So much so that phosphatases were at some point called in this beautiful review by David Brottingen, who studied phosphatases for many more years than I have. He called phosphatases the ugly ducklings of cell signaling. That gives you an idea of how bad it was for people to actually even consider studying this class of enzyme. Yeah, so this review is a very interesting one to read. So I really want to take you with me to take a step back now and think about this notion of selectivity. We mammals have evolved through billion years of evolution, and everything our cells and organisms do has been evolutionarily optimized. I've told you that protein phosphorylation is a very important process that controls the activity of many proteins in our cells. And the dogma at some point was that protein phosphorylation, which is controlled through the antagonistic action of kinases and phosphatases, is on one way selective and on the other way unselective. That doesn't make sense to me, particularly if you think about the fact that any imbalance in protein phosphorylation is highly deleterious. And to illustrate that, I can bring the example of cancer, for example, where abnormal protein phosphorylation is devastating. It needs to be tightly controlled, and so we need selective dephosphorylation as much as we need selective phosphorylation. So I'm going to solve this puzzle for you, and the answer to this problem is on this slide. Remember, the highly purified phosphatase was naked and stripped off its cellular cofactor, but in the cell, PP1 is bound to one amongst an array of up to 100 cellular proteins that actually give rise to selectivity. So phosphatases do not act alone, they don't act solo, they are bound to cellular proteins, and it is the complex formed by the phosphatase and the non-catalytic subunit that is highly selective. And so this is very important because here we have not one phosphatase PP1, but actually three, because there's three genes encoding PP1 in mammals. But these three catalytic subunits can be bound to one amongst 100 of diverse cellular proteins. So in this way, there's a large repertoire of phosphatases. And this holds true for virtually all phosphatases of the PPP family. So here we have solved a selectivity problem. We have at least as many phosphatases as we have kinases, and I will argue that phosphatases are also very selective. So I will now spend the rest of this talk going through the mechanism by which selectivity is actually achieved in phosphatases, and I'll start with the historical findings. So remember, initially the idea was PP1 is not really selective. When highly purified, it can dephosphorylate many substrates. Then came the discovery of inhibitor 1, and so the idea was, well, phosphatase in the cell needs to be sequestered, bound to an inhibitor to prevent it from engaging in non-selective dephosphorylation. So that's one way to control selectivity, but I'd argue that this is not enough to encode the tight selectivity that we need for our cells and organisms to function properly. A second level of selectivity came with the discovery of targeting subunits, and the first one came again from studying glycogen metabolism. Here comes the discovery of, by the Cohens, of a protein that they named GM. G stands for glycogen and M from muscle because it was purified from muscle. And this targeting subunit has the property to bind PP1 on one hand and glycogen on the other. And in this way, it concentrates PP1 in a glycogen granule. And so in this way, selectivity is thought to be improved by increasing the local concentration of PP1 in around glycogen where enzymes of glycogen metabolism are also localized. But whilst this is one way to increase selectivity, it may not be good enough to offer the tight control that one needs for glycogen metabolism. This is a very important physiologically controlled event, and misregulation of glycogen metabolism can cause diabetes, for example. So it may be beneficial to have an additional level of regulation here. And I would suggest that perhaps one needs also to precisely target the phosphatase to the enzymes that need to be dephosphorylated at any given time. So a third idea and model to account for the selectivity of phosphatases come from a study in my own lab of two serine threonine phosphatases, and we found that the non-catalytic subunit is actually not only recruiting the catalytic subunit PP1, but also very important in providing a high affinity binding site for the substrate. And this led me to propose that PP1 phosphatases are actually split enzymes because they are composed of two components, and each of them are essential. You need the two components to be together to form a functional holoenzyme. You need the catalytic subunit, and that's the one that cleaves off the phosphate, but that's of no use if the substrate is not correctly brought to the phosphatase, and that's the function of the non-catalytic subunit. So in this talk, I started giving you a historical overview of the discovery of phosphatases and how the notion that phosphatases were unselective arose. I've ended by telling you that actually phosphatases are exquisitely selective, and this is because they are composed of these two components, the common catalytic subunit bound to a selectivity factor, the non-catalytic subunit, which actually is important because it serves as a high affinity binding site for the substrate. So because phosphatases were thought to be unselective, they were also thought to be undruggable, and in the 90s, inhibitors were developed that inhibit PP1, and it was found that these inhibitors are actually not selective, because if you inhibit PP1, which is a component of hundreds of enzymes in the cell, it's not one enzyme that you inhibit, but actually hundreds of enzymes. And that's actually toxic and not very useful, so that led pharmaceutical industry to stop considering phosphatases, although they had recognized that protein phosphorylation was very important for drug discovery, and indeed kinases have been very, very popular, amongst the most popular drug targets in pharmaceutical industry. Recently, in my lab, we found that we can selectively inhibit a phosphatase by targeting its regulatory subunit, and we think that with this paradigm, we can expand and inhibit other enzymes. So if you want to learn about this, how we discovered how to inhibit phosphatase, and if you want to hear about assays and methods to identify selective phosphatase inhibitors, I'll invite you to join me and listen to the second and the third talk of this series. Thank you very much.